The LAC operon is an important DNA sequence in many different prokaryotic species that encodes for the proteins that are necessary to break down lactose. The way that it works is by RNA polymerase binding to a promoter region upstream of the LAC operon, which will then go on to transcribe the LAC-I gene, which encodes for a repressor protein that will bind onto the operator region of the LAC operon. Now, when RNA polymerase binds onto the LAC-P promoter, it cannot go through and transcribe the rest of the LAC operon. This is known as negative control because the regulator protein is bound to the DNA and stops transcription. But when lactose is present in the system, allolactose will also be present, and allolactose will bind to the allosteric site of the repressor and cause conformational changes that will no longer allow it to bind to the operator. RNA polymerase can now go through and transcribe the rest of the DNA. This is inducible control because when lactose is present in the system, it induces transcription. The LAC-Z gene encodes for beta-galactosidase, which converts lactose into glucose and galactose, which can be easily metabolized by the bacteria. It also turns lactose into allolactose, which, as we talked about, is used in inducible control. The LAC-Y gene encodes for permease, which is used in the cell membrane because the cell membrane usually can't be penetrated by lactose. But when permease is in the cell membrane, lactose can easily go through and be part of the system. The previous LAC operon that I described was a wild-type LAC operon. However, there can be mutations in a LAC operon, and the single cell can contain multiple LAC operons. For example, a chromosomal LAC operon and a plasmid one. We'll start by analyzing the chromosomal LAC operon of this cell. This LAC operon has a mutation in the LAC-I gene that will produce a super repressor, which means this repressor does not have a binding site for allolactose, which means if bound to the operator, it won't be able to come off. However, the operator for this LAC operon has a mutation making it constitutive, which means that no repressor can bind to it. But this LAC operon has a mutation in the promoter, which makes RNA polymerase unable to bind, rendering it virtually useless. As for the plasmid, there's a mutation in the LAC-I gene, making it so no repressor is produced, and there's a normal promoter. But due to the super repressor being made from the other LAC operon on the chromosome, it's going to bind onto that operator and be stuck on there, and RNA polymerase won't be able to go through and transcribe these genes. And even if it could, there's a mutation in the LAC-Z gene which would cause there to be dysfunctional beta-galactosidase. So this LAC operon is pretty much useless too.